Good evening and welcome to Dreamy Academy. Tonight we begin a journey not just through concepts, but through the very fabric of existence, seeking the quiet wonders that hum beneath the surface of our world. As we begin, I'd love to know where you're watching from, your city, your country, perhaps even what the night sky looks like outside your window. Is it clear and star-dusted, or softly veiled by clouds? If you find peace and fascination in these explorations of science, please take a moment to like the video and subscribe. It truly helps this little corner of quiet curiosity grow. To those already subscribed, thank you sincerely for being part of this slow and thoughtful adventure, allowing these whispers of the universe to drift into your quiet hours. Right now, as you listen, forces older than mountains are shaping the atoms in your bones, in the air you breathe, in the very floor beneath your feet. Every breath you take contains atoms that have journeyed through cosmic dust, through ancient seas, through dinosaur lungs, and perhaps even through the heart of a star. You are, in this profound sense, a temporary arrangement of universal matter, caught in a delicate, intricate dance. We often imagine life as something separate from the non-living world, a vibrant, complex exception to the inertness of rock and water. But what if we were to look closer, to truly understand that the boundary is far more permeable than we think? What if life, in all its astonishing complexity, is simply chemistry that learned to remember itself, to replicate, to evolve? This is the story we will unfold together. The story of biochemistry, the hidden language that binds the living world to the physical universe. It's a tale told not in grand pronouncements, but in the patient, precise interactions of atoms and molecules, a narrative of such profound beauty and elegance that it can lull the mind into a state of deep wonder, inviting not just understanding, but a quiet, satisfying surrender to the interwoven tapestry of existence. Consider for a moment the ordinary miracle of a single breath. It is a simple act, perhaps the most fundamental rhythm of your being. Air flows in, air flows out, yet within that seemingly effortless exchange, a universe of silent, ceaseless activity unfolds. Billions upon billions of tiny particles, far too small to ever truly see, are engaging in a meticulously choreographed dance, exchanging partners, breaking old ties, forming new ones, all to sustain the warmth and light of your life. This isn't magic, though it feels like it. This is the bedrock of biochemistry, the fundamental architecture of atoms, and the invisible yet immensely powerful bonds that hold them together. Truly appreciate this quiet marvel, we must first step outside our human scale of perception and journey inward, past the visible, past the microscopic, into a realm where certainty blurs into probability. Let us begin by considering the atom itself, the fundamental building block of everything you know, everything you are. Picture, if you will, not a solid, irreducible speck, but a vast, mostly empty space. At its heart, a tiny, incredibly dense nucleus, a tightly packed cluster of protons and neutrons. These protons, each carrying a positive electrical charge, define the atom's identity. 1 proton means hydrogen, 6 means carbon, 8 means oxygen. It is a simple, elegant system of classification. Around this nucleus, like a shimmering, probabilistic cloud, electrons drift and dance. These tiny particles carry a negative charge, an equal and opposite counterpoint to the protons. They don't orbit in neat planetary paths, as early models might have suggested. Instead, they exist in regions of probability, fuzzy energetic shells where they are most likely to be found. The atom, then, is less like a miniature solar system and more like a whisper of energy, a potentiality with an incredibly dense core and a vast empty expanse where electrons flicker in and out of definable location. The nucleus, though containing almost all the atom's mass, over 99.9%, occupies only a minuscule fraction of its volume, perhaps 1 part in 10,000 trillion. This means that the chair you sit upon, the air you breathe, and your very own hand are in essence mostly emptiness, a beautiful paradox that humbles our sense of solidity. It is in this elegant dance of electrons that the story of life truly begins. Atoms, it turns out, are not content in isolation. They yearn for a state of stability, a sense of energetic balance, and they achieve this balance by interacting with other atoms, by sharing or exchanging their outermost electrons. This drive for equilibrium is the engine of all chemistry and thus the engine of life itself. These interactions, these energetic handshakes and embraces, are what we call chemical bonds. They are the invisible threads that weave the universe into form, from the simplest molecule of water to the most intricate protein. Imagine 2 hydrogen atoms, each with a single proton and a single electron. They are incomplete, restless. Now introduce an oxygen atom, with its 8 protons and 8 electrons, 2 in an inner shell and 6 in an outer shell, seeking 2 more to achieve balance. What happens? The hydrogen atoms approach the oxygen, and in an act of profound energetic grace they share their electrons. Each hydrogen contributes 1, and the oxygen contributes 1 to each bond, creating a stable shared partnership. This is a covalent bond, a strong intimate connection where electrons are held in common, binding the atoms together into a single cohesive unit, a molecule of water. It is a bond that requires energy to break and releases energy when formed, a fundamental principle of all chemical reactions. Consider too another kind of interaction, equally vital: the ionic bond. Here, the exchange is not a sharing but a transfer. Picture a sodium atom with 1 restless electron in its outer shell, and a chlorine atom needing just 1 more electron to feel complete. Rather than sharing, the sodium generously donates its electron to the chlorine. Sodium, now lacking a negative charge, becomes a positive ion. Chlorine, gaining a negative charge, becomes a negative ion. And like 2 magnets, these oppositely charged ions are drawn together by powerful electrostatic forces, forming a crystal of sodium chloride, common table salt. This bond too holds potential energy, a quiet strength waiting to be unleashed or transformed. These bonds, covalent and ionic, are the alphabet of life's language. They dictate the shapes molecules can take, the ways they can interact, the specific functions they can perform. The precise angles at which atoms bond, for instance, are not random. They are governed by the quantum mechanics of electron clouds, creating specific geometric arrangements. A water molecule, for example, is not linear. Its 2 hydrogen atoms are bent at an angle of roughly 104.5 degrees relative to the oxygen atom. This seemingly small detail has profound consequences, making water a polar molecule with a slightly negative side and a slightly positive side, capable of dissolving a vast array of substances and forming intricate networks with other water molecules. This unique architecture is why ice floats, why lakes don't freeze solid from the bottom up, and why life, as we know it, could emerge and persist. From these simple, elegant rules, the desire of electrons for stability, the formation of bonds, the resulting molecular shapes, an astonishing complexity begins to unfold. It is a complexity built layer by layer, like a geological record, each interaction laying the groundwork for the next. The universe, it seems, has a deep tendency towards building, towards arranging its fundamental particles into ever more intricate and fascinating patterns. These patterns store energy, transfer information, and eventually begin to exhibit the qualities we recognize as life. The very atoms that once danced in the heart of a star now dance within your cells, held together by the same fundamental forces engaged in the timeless quest for energetic equilibrium. We've seen how individual atoms, through their electron shells, are compelled to connect, forming the foundational bonds that create molecules. But how do these initial simple structures begin to assemble into the vast, intricate molecular machinery that defines a living cell? How does the chemistry of water, for instance, pave the way for the chemistry of DNA or the chemistry of thought? That question invites us to consider not just individual bonds, but the larger architecture they build and the unique properties that emerge when molecules begin to interact with each other in subtle yet powerful ways. This is where the true elegance of biochemistry begins to reveal itself, a testament to the universe's capacity for self-organization. We left off, didn't we, with a question echoing in the quiet halls of our dreamy academy. How do these fundamental atomic attractions, these simple dances of electrons forming covalent and ionic bonds, scale up? How do these precise molecular geometries, like water's bent polar embrace, give rise to the vast, intricate, self-organizing molecular machinery that defines a living cell? It is a profound leap from the whisper of an atom to the symphony of a cell, and yet the universe, in its infinite elegance, has provided the answer. We might imagine, perhaps, that once molecules are formed, their work is done, their identities sealed. But in the ceaseless, vibrant realm of biochemistry, nothing is ever truly static or isolated. Consider for a moment the very air you breathe, the water you drink, the cells that compose your being. They are not merely collections of individual molecules floating aimlessly. No, they are vast, intricate tapestries woven from countless molecular interactions, a bustling metropolis where every resident molecule is constantly engaging in energetic handshakes, fleeting embraces, and subtle pushes and pulls with its neighbours. These aren't the strong, unwavering commitments of the covalent or ionic bonds we explored, which define a molecule's very identity. Instead, we are now turning our gaze to the quieter, gentler forces, the intermolecular forces. These are the subtle attractions and repulsions between molecules, the silent conversations that dictate how they arrange themselves, recognize one another, and ultimately how they cooperate to build the grand architecture of life. Individually, they are mere whispers. Collectively, however, they orchestrate the most magnificent ballets. Let us begin with perhaps the most celebrated and ubiquitous of these forces, one that is utterly indispensable to life as we know it, the hydrogen bond. Recall our friend, the water molecule, a small marvel of a molecule with its oxygen atom greedily pulling electrons, leaving its two hydrogen atoms with a slight positive charge and itself with a slight negative charge. This polarity is the key. When one water molecule with its partially positive hydrogen comes near another water molecule with its partially negative oxygen, there's an undeniable gentle tug. This attraction, this subtle yet persistent yearning for connection, is a hydrogen bond. Imagine a microscopic dance floor teeming with water molecules. Each oxygen, holding its two hydrogen partners, is like a dancer with two outstretched hands, the partially positive hydrogens, and a partially negative back. When one dancer's outstretched hand brushes against another's back, a temporary yet significant connection is made. These bonds are not as strong as the covalent bonds holding the hydrogen and oxygen within a single water molecule, but they are strong enough to create a complex dynamic network. Each water molecule can form up to 4 hydrogen bonds with its neighbours, linking them together in a shimmering, ever-shifting lattice. This is why water has such extraordinary properties. Its high boiling point, its remarkable ability to dissolve so many substances, and crucially, why ice floats, a crystalline structure of hydrogen bonds that holds molecules further apart than in liquid water. Without this elegant property, the very oceans would freeze from the bottom up, extinguishing life before it could begin. But hydrogen bonds are not exclusive to water. Any molecule possessing a hydrogen atom covalently bonded to a highly electronegative atom, most commonly oxygen, nitrogen, or fluorine, can participate in these vital energetic handshakes. Consider the vast complex molecules within your own cells. Proteins, nucleic acids like DNA. Their intricate three-dimensional shapes, which dictate their very function, are meticulously sculpted and held together by thousands upon thousands of these hydrogen bonds. They are the invisible threads that pull a long, floppy chain of amino acids into a precisely folded enzyme, or zip together the 2 strands of DNA into its iconic double helix, encoding the very blueprint of life. Without them, these crucial macromolecules would simply unravel, dissolving into formless chaos. Beyond the elegant specificity of hydrogen bonds, other intermolecular forces play their part in this grand molecular architecture. We encounter the spectrum of van der Waals forces, named after the Dutch physicist Johannes Diederik van der Waals, who first proposed their existence. These are even more subtle, more fleeting, yet profoundly important. First, there are the dipole-dipole interactions. These occur between 2 polar molecules that, like water, possess a permanent uneven distribution of charge. If you have 2 tiny molecular magnets, they will naturally orient themselves to attract one another, positive end to negative end. These attractions are stronger than the transient forces we'll discuss next, but still weaker than hydrogen bonds. They contribute to the solubility of polar substances in polar solvents and influence how molecules pack together. Then we delve into the most ephemeral yet universally present of these forces, the London dispersion forces, sometimes simply called dispersion forces. These occur between all molecules, whether they are polar or nonpolar. How can this be? Imagine, if you will, the electron cloud of an atom or molecule. It's not a static, perfectly balanced sphere, but rather a shimmering, probabilistic cloud where electrons are constantly in motion. For a fleeting instant, purely by chance, more electrons might gather on one side of an atom than the other, creating a temporary instantaneous dipole. This momentary imbalance can then induce a corresponding dipole in a neighbouring molecule, like a tiny ripple effect. These 2 transient dipoles then attract each other for a fraction of a second. Though individually incredibly weak and fleeting, when we consider a large molecule with many atoms, or vast numbers of molecules packed closely together, these forces add up significantly. They are the primary forces holding together nonpolar molecules, like the long hydrocarbon chains in fats and oils, allowing them to coalesce and form structures. They are the glue for the seemingly inert. And then we arrive at a phenomenon that is less a direct attractive force and more a profound consequence of water's nature, the hydrophobic effect. This is a concept of immense importance in biochemistry. Imagine trying to mix oil and water. They famously don't mix, do they? This isn't because the oil molecules repel the water molecules in an active sense. Rather, it's because the water molecules prefer to hydrogen bond with each other. When nonpolar molecules like oil are introduced into water, they disrupt water's beautiful, extensive hydrogen bond network. To minimize this disruption, The water molecules essentially push the nonpolar molecules together, forcing them to coalesce. It's as if the water molecules are saying, we'd rather spend our energy forming stable, satisfying hydrogen bonds with our own kind. So you nonpolar guests, please gather amongst yourselves and leave us to our preferred interactions. This squeezing out of nonpolar molecules by water is the hydrophobic effect, and it is a major driving force in the self-assembly of biological structures. It is what causes lipids to form cell membranes, creating the essential boundaries that define a cell. It is what drives the interior folding of proteins, bearing their non-polar amino acid side chains away from the watery cellular environment, allowing the polar and charged groups to remain on the surface, ready to interact with water and other molecules. Here we stand, gazing upon this exquisite symphony of intermolecular forces. Each individual note, a hydrogen bond, a dipole-dipole attraction, a fleeting dispersion force, is weak, easily broken and reformed. But it is precisely this weakness, this transience, that lends them their power. They allow for dynamic interactions, for molecules to bind and release, to shift and reconfigure, which is absolutely essential for the constant activity of life. Think of an enzyme binding to its substrate, performing its catalytic magic, and then releasing the product. This molecular recognition and action is largely orchestrated by these weak, reversible, energetic handshakes. These forces are the silent architects that transform a collection of individual molecules into organized, functional structures. They are the reason why water, the solvent of life, is not merely a passive medium, but an active participant. Its hydrogen bonding network and its hydrophobic effect dictating the very shape and behavior of the molecules dissolved within it. It is the stage upon which life's drama unfolds, and the stage itself plays a starring role. As we move forward, we will see how these fundamental principles, the yearning for stability, the precise geometries dictated by covalent bonds, and the intricate dance of intermolecular forces, build upon one another. They are the foundational grammar, the very alphabet of life's language. From these seemingly simple rules emerge the colossal complex macromolecules, the proteins with their myriad functions, the nucleic acids storing and transmitting genetic information, the carbohydrates providing energy and structure, and the lipids forming barriers and signaling molecules. These are the intricate sentences and paragraphs that tell the story of life, all constructed from the humble letters of atomic interactions and the subtle punctuation of intermolecular forces. The journey, dear friends, is only just beginning. We have peeled back another layer of the universe's quiet wonders, revealing how elegant simplicity gives rise to astonishing complexity. But how do these larger molecules, these macromolecules, then assemble into the intricate machinery of a living cell? How do they communicate, replicate, and evolve? These are the questions that beckon us onward, further into the heart of biochemistry's profound beauty. We have journeyed through the subtle dance of atomic attraction, explored the profound implications of shared electrons and transferred charges, and marveled at the ephemeral yet mighty whispers of intermolecular forces. We have watched as water, the very solvent of life, orchestrates its grand hydrophobic ballet, urging disparate molecules into ordered structures. But the question lingers, echoing in the quiet chambers of our inquiry. How do these fundamental interactions translate into the colossal, intricate architectures that define life? How do these larger molecules, these macromolecules, assemble, communicate, replicate, and evolve within the vibrant, bustling theatre of a living cell? Today, we turn our gaze from the forces that bind to the magnificent structures they erect. We step into the grand hall where the silent architects have laid down their tools, revealing the very foundations upon which all life is built. We speak, of course, of the four great classes of macromolecules: carbohydrates, lipids, proteins, and nucleic acids, each a testament to nature's ingenuity, each a chapter in the unfolding epic of existence. Consider, if you will, the principle of elegant repetition. Just as a master weaver constructs a complex tapestry from countless individual threads, so too does life craft its grandest molecules from simpler recurring units. Most of these macromolecules are polymers, vast chains woven from smaller, identical or near-identical building blocks known as monomers. It is a symphony of unity and diversity, where a limited set of individual notes can give rise to an infinite array of melodies. Let us begin with the sweet essence of life itself, the carbohydrates. From the simple sweetness that graces our tongues to the sturdy scaffolding that holds giant trees aloft, carbohydrates are everywhere, a fundamental expression of solar energy captured and stored. Their monomers are the monosaccharides, simple sugars like the ubiquitous glucose, a molecule so perfectly balanced, so readily available, that it serves as the universal currency of immediate energy for nearly all life forms. Imagine glucose as a small 6-carbon ring, a hexagonal jewel brimming with potential energy, patiently awaiting its call to action. These monosaccharides, these individual sugars, are not content to exist in isolation. They yearn for connection, reaching out through energetic handshakes to form glycosidic linkages, covalent bonds that join them into longer, more complex chains. When two join, we have a disaccharide like the familiar sucrose, the sugar we sweeten our tea with. When many join, they form polysaccharides, truly immense structures that serve a multitude of roles. Think of starch, the branched energy reservoir in plants, or glycogen, its animal counterpart, a molecular pantry in our muscles and liver, ready to release glucose at a moment's notice. Or consider cellulose, a linear, rigid polysaccharide that forms the cell walls of plants, providing structure and strength, a silent, woody backbone that allows life to reach for the sky. These are not merely inert molecules, they are dynamic, ever-ready reservoirs, structural engineers, and even the very markers on cell surfaces that allow cells to recognize friend from foe. They are the initial spark, the immediate fuel, and the enduring framework, all spun from the same simple sweet thread. Next, we turn our attention to a class of molecules that often shies away from water's embrace, preferring instead the company of their own kind, the lipids. Unlike carbohydrates, proteins, and nucleic acids, lipids are not typically polymers built from repeating monomeric units. Instead, they are a diverse group united by a singular defining characteristic: their profound hydrophobicity. They are the oil in water, the immiscible boundary, and in this very aversion to water, they find their most profound purpose. Picture the humble triglyceride, a fat molecule composed of a glycerol backbone to which 3 long hydrocarbon chains, the fatty acids, are attached. These long, non-polar tails recoil from water, clustering together to minimize their disruptive effect on water's intricate hydrogen bond network. This inherent tendency of lipids to self-associate in aqueous environments is not a flaw, but a design principle of unparalleled elegance. It is the very engine behind the formation of cellular membranes, those shimmering fluid boundaries that define the inside from the outside, the self from the not-self. Phospholipids, with their dual nature, a hydrophilic head that loves water and 2 hydrophobic tails that shun it, are the master builders of these membranes. They spontaneously arrange into bilayers, forming the delicate, dynamic walls of every cell, every organelle, a testament to the power of the hydrophobic effect we explored in our last chapter. Beyond structural roles, lipids are also incredible energy stores, packing more than twice the energy per gram compared to carbohydrates. They are the silent guardians, the energetic reservoirs and the messengers, like the steroid hormones, that travel through our bodies, whispering instructions from one cell to another. They are the boundaries and the signals, all born from their inherent reluctance to dissolve. Now, let us ascend to the most versatile and astonishing class of macromolecules, the proteins. If carbohydrates are the fuel and structure, and lipids the boundaries and long-term stores, then proteins are the molecular artisans, the cellular symphony conductors, the tireless workhorses that perform virtually every task within a living organism. They are the enzymes that catalyze reactions with breathtaking speed and specificity, the transporters that shuttle molecules across membranes, the antibodies that defend against invaders, the structural components that give cells their shape, and the motors that power movement. Their diversity is staggering, their functions seemingly endless. The monomers of proteins are the amino acids, a mere 20 distinct types, each a unique letter in life's profound alphabet. Each amino acid possesses a central carbon atom bonded to an amino group, a carboxyl group, a hydrogen atom, and a variable side chain, or R group, which gives each amino acid its unique chemical personality, whether it's polar, nonpolar, acidic, or basic. Imagine these 20 letters, each with its own subtle character, lined up in a specific sequence. Through the formation of peptide bonds, another strong covalent linkage, forged between the carboxyl group of one amino acid and the amino group of another. These amino acids are strung together into long, unbranched chains called polypeptides. But the magic of proteins truly begins after this chain is formed. The precise linear sequence of amino acids, the primary structure, dictates how this chain will fold into a unique and intricate three-dimensional shape. Driven by the very intermolecular forces we discussed, hydrogen bonds, van der Waals interactions, ionic bonds between charged R groups, and even covalent disulfide bridges, the polypeptide contorts and coils, bends and loops, until it achieves its functional form. This specific, often incredibly complex three-dimensional conformation is absolutely critical to a protein's function. A slight change in shape can render it useless, or even harmful. Proteins are the ultimate expression of form-meeting function, where the delicate dance of atomic attraction sculpts a molecular machine capable of wonders. They are the very essence of cellular action, the agents of change, and the embodiment of life's dynamic purpose. Finally, we arrive at the very heart of heredity, the blueprint of life itself, the nucleic acids. These are the molecules that carry the genetic information, the ancient script that dictates the construction of every protein, every cell, every organism. They are the keepers of continuity, ensuring that life's intricate instructions are passed faithfully from one generation to the next. The monomers of nucleic acids are nucleotides, each a complex unit consisting of 3 parts: a 5-carbon sugar, deoxyribose in DNA, ribose in RNA, a phosphate group, and a nitrogenous base. It is these nitrogenous bases, adenine, A, guanine, G, cytosine, C, and thymine, T in DNA, with uracil, U, replacing thymine in RNA, that form the informational alphabet. Like beads on a string, nucleotides are linked together by phosphodiester bonds, strong covalent bonds formed between the phosphate group of one nucleotide and the sugar of another. This creates a long sugar-phosphate backbone with the nitrogenous bases projecting inwards. In DNA, two such polynucleotide strands entwine to form the iconic double helix, a structure of unparalleled elegance and profound significance. The two strands are held together by hydrogen bonds between complementary base pairs, A always pairing with T and G always pairing with C. This precise pairing is the foundation of genetic information storage and replication, allowing the molecule to be faithfully copied and its information decoded. RNA, typically a single-stranded molecule, plays a crucial role in translating this genetic information into the language of proteins. Nucleic acids are not just molecules, they are the whispers of eternity, the ancient script of creation, the very instructions that allow life to remember itself, to replicate, to adapt, and to evolve. They are the architects of information, the librarians of life's grand narrative. As we survey these four magnificent classes of macromolecules, carbohydrates, lipids, proteins, and nucleic acids, we begin to grasp the sheer scope of nature's constructive genius. From the simple yearning of atoms for stability, guided by the subtle forces of intermolecular attraction, we witness the emergence of these complex purposeful structures. Each class plays its vital, indispensable role, contributing to the grand interconnected tapestry of life. They are the bricks and mortar, the girders and wires, the very operating system of every living entity. But understanding their individual forms is only the first step. The true marvel lies in their choreographed interactions, their ceaseless collaboration within the bustling confines of a cell. How do these molecular giants, once assembled, perform their intricate tasks? How do they recognize each other, bind with exquisite specificity, and catalyze the myriad reactions essential for life? This is where the principles of structure and function truly converge, a journey into the dynamic world of molecular machinery that awaits our exploration. For now, let us simply stand in awe of these silent architects, these fundamental building blocks, each a masterpiece, each a testament to the profound beauty of biochemistry. We left our journey at a precipice, gazing upon the magnificent architecture of macromolecules. We had seen how the subtle whispers of intermolecular forces coaxed simple monomers into the grand narratives of carbohydrates, lipids, proteins, and nucleic acids. We understood their structure, their potential, their very essence. But a profound question lingered, a quiet echo in the hallowed halls of our dreamy academy. How do these assembled titans, these intricate molecular machines, actually perform their tasks? How do they recognize each other, engage in the precise ballets of interaction, and catalyze the myriad reactions essential for the vibrant, pulsating reality we call life? Consider for a moment the cell not as a static museum of perfect forms, but as a bustling, vibrant city, alive with the ceaseless hum of activity. Within its shimmering boundaries, countless chemical reactions unfold every second, a dizzying, intricate dance of making and breaking bonds, transforming one molecule into another. It is a symphony of transformation, a continuous act of molecular alchemy. Yet left to their own devices, many of these vital reactions would proceed at a pace so glacial, so imperceptible, that life as we know it would simply cease to be. The universe, in its grand design, often sets formidable barriers, even for processes that are thermodynamically favoured. This brings us to one of nature's most elegant solutions, a concept that sits at the very heart of biological dynamism: activation energy. Imagine standing at the foot of a majestic mountain pass, on one side a verdant valley, on the other an equally inviting landscape. The journey across the pass is energetically favourable. You'll expend energy to climb, but once at the summit, the descent is effortless, leading to a state of lower potential. Yet, the climb itself is the barrier, the initial input of effort required to reach the peak. In the molecular world, this climb is the activation energy, the minimum energy required to initiate a chemical reaction, to strain existing bonds, to bring reactants into the precise orientation needed for transformation. Without a way to surmount this energetic peak, even the most eager molecules might simply linger, perpetually on the verge of change, but never quite achieving it. And here, dear explorers, we encounter the unsung heroes, the tireless facilitators, the very alchemists of life. Enzymes. These are the cellular architects of speed, the master orchestrators that ensure life's symphony plays not only beautifully but also on time. Almost invariably, enzymes are proteins, those remarkably versatile macromolecules we explored in our last chapter, whose exquisite three-dimensional shapes are sculpted by the subtle interplay of amino acid sequences and intermolecular forces. What is their profound secret? Enzymes do not alter the fundamental thermodynamics of a reaction. They do not change whether a reaction is ultimately favourable or unfavourable. Instead, they operate with a quiet, potent grace, like a skilled guide finding a hidden, gentler path over that formidable mountain pass. They lower the activation energy. They do this by providing an alternative reaction pathway, one that requires less initial energetic input, thereby accelerating the rate at which equilibrium is reached, often by factors of millions or even billions. They are the ultimate efficiency experts, allowing the cell to perform its intricate chemical transformations with breathtaking speed and precision, all within the gentle, temperate conditions of living systems. The magic of an enzyme lies in its exquisite specificity. Each enzyme, a miniature molecular artisan, is typically designed to interact with only one or a very small set of specific molecules, known as its TUI substrates. This specificity arises from the enzyme's unique three-dimensional structure, particularly a specialized region called the active site. Picture this active site as a precisely sculpted pocket or groove on the enzyme's surface, a molecular embrace perfectly contoured to cradle its specific substrate. It's not merely a lock and key fit, though that analogy offers a useful starting point. Rather, it's more akin to a gentle, evolving handshake. When a substrate molecule diffuses into proximity, the enzyme doesn't just passively accept it. Instead, the interaction induces subtle conformational changes in both the enzyme and the substrate, a dynamic process known as the induced fit model. The active site molds itself around the substrate like a glove conforming to a hand, optimizing the fit and bringing key catalytic residues into precise alignment. This intimate embrace is stabilized by a fleeting network of weak, non-covalent interactions, hydrogen bonds, ionic interactions, van der Waals forces, the very same forces we marvelled at in Chapter 2, now deployed with masterful intention. Once nestled within the active site, the enzyme employs a variety of ingenious strategies to lower the activation energy. It might orient the substrates perfectly, bringing their reactive groups into optimal proximity and alignment. It might subtly strain the existing bonds within the substrate, weakening them and making them more susceptible to breaking. It might create a unique microenvironment within the active site, perhaps a pocket of specific pH or polarity, that favours the transition state of the reaction. It can even participate directly in the chemistry, temporarily forming covalent bonds with the substrate before releasing it, regenerated and ready for its next catalytic cycle. Consider the enzyme hexokinase, which initiates the first step of glucose metabolism by adding a phosphate group to glucose. When glucose binds to hexokinase, the enzyme undergoes a significant conformational change, effectively closing around the glucose molecule. This not only excludes water from the active site, preventing unwanted side reactions, but also brings ATP, the phosphate donor, into the perfect position to transfer its phosphate to glucose. It's a testament to nature's engineering brilliance, a precise dynamic interaction leading to a swift, targeted chemical transformation. But the story of enzymes doesn't end with their catalytic prowess. For life to be orderly and responsive, these molecular alchemists cannot simply operate at full throttle all the time. Their activity must be meticulously controlled, finely tuned to the ever-changing needs of the cell and the organism. This regulation is another marvel of biochemical sophistication, ensuring that resources are not wasted and metabolic pathways proceed in a harmonious, coordinated fashion. One primary mode of control is allosteric regulation. Imagine an enzyme with not just one active site, but also another, distinct site, an allosteric site. When a regulatory molecule binds to this allosteric site, it induces a conformational change in the enzyme's overall structure, subtly altering the shape of the active site. This change can either enhance the enzyme's activity, allosteric activation, or inhibit it, allosteric inhibition. It's like a remote control, allowing signals from other parts of the cell to modulate the enzyme's function without directly competing for the active site. This allows for rapid, reversible fine-tuning of metabolic fluxes. Another crucial regulatory mechanism involves covalent modification, often through the addition or removal of small chemical groups. The most ubiquitous example is phosphorylation, where a phosphate group is covalently attached to a specific amino acid residue, like serine, threonine, or tyrosine, on the enzyme. This addition, often catalyzed by other enzymes called kinases, can dramatically alter the enzyme's conformation and consequently its activity, switching it on or off or modulating its efficiency. The removal of this phosphate group by phosphatases reverses the effect, creating a dynamic switch that allows for complex signaling networks within the cell. Perhaps the most elegant example of enzyme regulation in action is feedback inhibition. Envision a metabolic pathway as a production line, where a series of enzymes sequentially transform a starting molecule into a final product. If the cell has produced enough of this final product, it makes sense to halt further production to conserve energy and resources. In feedback inhibition, the final product of a metabolic pathway acts as an allosteric inhibitor for an enzyme earlier in the pathway, often the very first enzyme. As the concentration of the product rises, it binds to the allosteric site of the initial enzyme, shutting down the entire pathway until more product is needed. It's a self-regulating loop, a testament to the inherent wisdom embedded within biological systems, preventing wasteful overproduction and ensuring a delicate balance. Thus, we see that the cell is not merely a bag of chemicals nor a haphazard collection of reactions. It is a meticulously choreographed symphony, a grand, dynamic dance orchestrated by the tireless work of enzymes. They are the conductors, the tempo setters, ensuring that every note is played precisely, every transformation occurs at the right moment, with astonishing speed and specificity. Without them, the intricate metabolic pathways, the breakdown of nutrients for energy, the synthesis of complex biomolecules, the detoxification of harmful substances, the replication of genetic material would grind to a halt. Life itself would be impossible. From the quiet yearning of atoms for stability, through the gentle embrace of intermolecular forces to the magnificent architectures of macromolecules, we now witness the active agents that bring these structures to life. Enzymes imbue the static potential of molecules with dynamic purpose, transforming the raw materials of existence into the vibrant, self-sustaining processes that define living organisms. They bridge the gap between structure and function, between possibility and reality. But how, you might wonder, does the cell power this ceaseless activity? How does it fuel the constant making and breaking of bonds, the constant maintenance of order in a universe that relentlessly tends towards disorder? If enzymes are the alchemists, what is their magical fuel? This profound question of energy, its capture, its storage and its release, will be our next grand exploration as we delve into the very energetics of life itself. For beneath the intricate dance of catalysis lies the fundamental truth. Life is a continuous struggle against entropy, powered by a constant flow of energy, a story we are only just beginning to unfold. And so we arrive at a most profound question, perhaps the most fundamental of all. How, dear friends, does the cell power this ceaseless enzymatic activity, this intricate dance of transformation we observed in our last contemplation? We have seen the silent architects, the delicate forces, the grand structures, and the tireless alchemists. But what fuels their work? What is the invisible current that animates this miniature universe within each cell? Today, we delve into the very breath of life itself, the energetic pulse that sustains all being. We speak of cellular energy, the magnificent choreography of how life captures, stores, and expends the vitality it needs to persist, to grow, to remember. Imagine, if you will, a vast, bustling city, alive with activity. Every shop, every factory, every home requires a constant supply of a universal currency to function. Without it, the city grinds to a halt. In the microscopic metropolis of the cell, this universal currency, this shimmering coin of life, is adenosine triphosphate, or ATP. Let us gaze upon this molecule, for it is a marvel of elegant design. At its heart lies adenosine, a combination of adenine, one of life's fundamental nitrogenous bases, and a 5-carbon sugar, ribose. But it is the 3 phosphate groups strung together like a string of pearls that hold the secret to ATP's power. Specifically, the bonds connecting the last 2 phosphate groups are often referred to as high-energy bonds. This is not to say they are inherently stronger than other covalent bonds, but rather that their hydrolysis, their breaking by the addition of water, releases a substantial amount of free energy. Think of ATP as a molecular spring coiled tightly, ready to release its stored tension, or perhaps as a tiny rechargeable battery. When the cell needs to perform work, to contract a muscle, to synthesize a protein, to transport a molecule against its concentration gradient, to activate an enzyme, it simply hydrolyzes the terminal phosphate bond. ATP sheds its outermost phosphate group, transforming into ADP, adenosine diphosphate, and an inorganic phosphate, pi. And in doing so, it releases a burst of energy. This energy, a whisper of the universe's own creative force, is then precisely channeled to drive an otherwise energetically unfavorable or endogonic reaction. It's an energetic handshake, a direct transfer of power from the liberated bond to the work at hand. But where does this ATP come from? How is this molecular battery recharged? This brings us to the grand narratives of metabolism, the sum total of all chemical reactions that occur within a living organism. Metabolism is a ceaseless, intricate dance of building up and breaking down, a testament to the dynamic equilibrium that defines life. For most of us, and indeed for most life on Earth, the journey begins with the breakdown of nutrient molecules, the carbohydrates, lipids, and even proteins we discussed earlier. This process, a series of interconnected pathways known as cellular respiration, is life's masterful way of extracting the stored energy from these fuel molecules and converting it into the usable form of ATP. Consider a molecule of glucose, a simple sugar, a small packet of solar energy captured long ago. Cellular respiration is a carefully orchestrated disassembly, a controlled burn that liberates this energy in small manageable steps, rather than in a single destructive explosion. The first act unfolds in the cytoplasm, the bustling inner fluid of the cell. This is glycolysis, a primeval pathway, ancient and universal, shared by nearly all life forms. Here, our 6-carbon glucose molecule is gently cleaved and rearranged into 2 molecules of pyruvate, 3-carbon compounds. A small but vital amount of ATP is generated directly during this stage, along with some electron carriers that will play a much larger role later. It's a modest beginning, a gentle stir of the energetic pot. The true energy harvest, however, takes place within the mitochondria, those magnificent organelles we often call the powerhouses of the cell. These are not mere containers. They are intricate, folded landscapes designed for maximum efficiency. Here, pyruvate is further processed and fed into the citric acid cycle, also known as the Krebs cycle. Imagine a molecular merry-go-round, where carbon atoms are systematically oxidized, releasing more carbon dioxide, the very breath we exhale, and, crucially, generating a wealth of high-energy electron carriers, primarily NADH and FADH2. These carriers are like tiny charged shuttles, laden with electrons eager to descend an energetic gradient. And descend they do in the final breathtaking act of cellular respiration, oxidative phosphorylation, which encompasses the whole electron transport chain, and chemiosmosis. This occurs on the inner mitochondrial membrane, a highly convoluted surface that maximizes area for this critical process. The electron carriers, NADH and FADH2, deliver their cargo of electrons to a series of protein complexes embedded within this membrane. As electrons are passed from one complex to the next, much like a bucket brigade, they gradually lose energy. This released energy is not wasted. It is used to pump protons, hydrogen ions, from the mitochondrial matrix into the intermembrane space, creating a steep electrochemical gradient, a reservoir of potential energy. Imagine a dam holding back a vast lake of water. The urge for these protons to flow back down their gradient to equalize the charge and concentration is immense, and they do so, but only through a special molecular turbine, ATP synthase. This remarkable enzyme, a true rotary motor at the molecular scale, harnesses the energy of this proton flow, the proton motive force, to catalyze the synthesis of vast quantities of ATP from ADP and inorganic phosphate. This is where the bulk of life's energy currency is minted, a testament to the elegant conversion of potential energy into chemical energy. And oxygen, the very air we breathe, serves as the final electron acceptor at the end of this chain, forming water. It is the quiet, essential partner in this grand, energetic symphony. But let us pause for a moment and widen our gaze. Where did the glucose, the initial fuel, acquire its stored energy? For many organisms, the answer lies in an even more profound process: photosynthesis. In the chloroplasts of plants, algae, and some bacteria, The radiant energy of sunlight is captured and transformed into chemical energy. Carbon dioxide from the atmosphere and water are ingeniously woven together to create glucose, the very sugar we just saw disassembled, and oxygen, the vital byproduct. This is the ultimate wellspring of nearly all life on Earth, a cosmic alchemy that translates stellar fire into the quiet, potent energy that fuels every cell, every thought, every movement. So we see a magnificent cycle, a continuous ebb and flow of energy. The sun powers photosynthesis, creating fuel. Respiration breaks down that fuel, creating ATP. And ATP powers the myriad functions of life, from the smallest enzymatic twitch to the most complex thought. This intricate network of energy generation and utilization is what we call metabolic pathways. These are not isolated reactions, but rather highly organized sequential steps, each catalyzed by a specific enzyme. We distinguish between two fundamental types of pathways. First, there are the catabolic pathways, those that break down complex molecules into simpler ones, releasing energy in the process. Cellular respiration, which we just explored, is the quintessential catabolic pathway. It's the process of dismantling, of liberating stored potential. Then there are the anabolic pathways, those that build complex molecules from simpler precursors requiring an input of energy. The synthesis of proteins from amino acids, of DNA from nucleotides, or of complex carbohydrates from simple sugars, these are all anabolic processes. They are the construction crews of the cell, building and repairing, creating the very structures of life. And here lies the elegant genius of ATP. It acts as the vital link, the universal connector between catabolism and anabolism. The energy released by catabolic pathways is captured in the high-energy bonds of ATP. This charged ATP then diffuses to where energy is needed, hydrolyzing to ADP and pi, and providing the necessary power for anabolic pathways to proceed. It is a continuous energy coupling, a perpetual dance of charging and discharging, ensuring that the cell always has the precise amount of energy available for its immediate needs. This entire metabolic network is not a chaotic free-for-all. It is meticulously regulated. The cell constantly monitors its energy levels, its nutrient availability, and its internal state. Enzymes, our alchemists from the last chapter, often sit at key control points within these pathways. Feedback inhibition, allosteric regulation, covalent modification, these mechanisms and many others ensure that metabolic pathways are switched on or off, sped up or slowed down, precisely when and where they are needed. It's a symphony of self-correction, maintaining the delicate balance, the homeostasis that defines a living system. From the sun's fiery heart to the gentle flutter of a hummingbird's wing, from the vastness of the cosmos to the intimate dance of molecules within a single cell, energy flows, transforms, and sustains. It is the silent, tireless engine of existence, the very pulse of life itself. We too are part of this grand energetic exchange, borrowing energy from the universe to fuel our own thoughts, our own understanding. We have now glimpsed the fundamental architecture of life, from the atomic embrace to the grand energy cycles that power every cellular breath. But how does this immense information, these blueprints for complexity, these instructions for the dance of life persist and perpetuate through generations? How does life remember itself? That, my friends, is the profound question we shall explore next, as we turn our gaze to the very language of inheritance. We have journeyed through the intricate dance of atoms, witnessed the subtle power of their bonds, marveled at the grand architectures of macromolecules, and understood the energetic handshakes that fuel every cellular breath. We've seen how life, in its infinite wisdom, harvests sunlight and breaks down nutrients to forge the universal currency of ATP, powering the ceaseless work within us. But as we stand at the precipice of this understanding, a profound question still hangs in the shimmering air of our dreamy academy, whispering through the quiet corridors of our contemplation. How does this immense information, these blueprints for complexity, these instructions for the dance of life persist and perpetuate through generations? How does life remember itself, not just for a moment, but across epochs, echoing its forms and functions from the first flicker of existence to the present day? Consider for a moment the sheer audacity of life. It's not enough to build a cell to power it, to make it move and respond. That cell must also know how to build another cell, an identical twin, a faithful replica, carrying within it the exact same instructions for its own intricate ballet. This is where we uncover one of life's most breathtaking secrets, its most enduring legacy, the molecule of inheritance, the grand archive of all biological possibility. We speak, of course, of DNA. Imagine, if you will, not a static blueprint tacked to a wall, but a living, breathing scroll of ancient wisdom, inscribed with the very language of being. This scroll is not merely a record, it is a dynamic instruction manual capable of self-replication and ceaseless expression. It holds the silent symphony that orchestrates the growth of a towering redwood, the delicate flutter of a butterfly's wing, the complex firing of your own neurons as you ponder these very words. What does this scroll look like, you ask? If we were to zoom in, past the cellular walls, beyond the nucleus, into the very heart of this informational marvel, we would find a structure of exquisite elegance, the double helix. Picture a grand spiral staircase coiling endlessly upwards, its railings crafted from alternating sugar and phosphate molecules. These form the sturdy, repeating backbone of each strand. But it is the steps of this staircase that hold the true magic, the coded messages of life. These steps are composed of 4 distinct chemical letters known as nitrogenous bases: adenine, A, guanine G, cytosine C, and thymine T. Each step is formed by 2 of these bases reaching across the central axis of the helix, performing a precise and unwavering dance of attraction. Adenine always, unfailingly, pairs with thymine, AT. Guanine always, without exception, pairs with cytosine, GC. This complementary base pairing is not merely a structural detail, it is the very soul of genetic information, the elegant simplicity that underpins life's boundless complexity. It is like a secret handshake, ensuring that each half of the staircase perfectly mirrors the other, holding within it the potential to rebuild the whole. This inherent complementarity is what makes the perpetuation of life's instructions possible. When a cell prepares to divide, when a new organism is to be formed, this grand scroll must be copied. And here we witness a marvel of molecular engineering known as DNA replication. Imagine our double helix, our spiral staircase gently unzipping itself, much like a zipper slowly opening. The hydrogen bonds that hold the complementary base pairs together, those quiet, energetic handshakes, softly release, separating the 2 strands. Each now exposed strand becomes a template, a guide for the creation of a brand new partner. As the strands unwind free-floating nucleotides, the individual sugar-phosphate base units begin to drift into place, guided by an inherent yearning for stability and the unerring rules of complementarity. An exposed A on the template strand will call out for a T and G for a C. And as these new partners find their match, a molecular architect, an enzyme called DNA polymerase, moves along the growing strand, deftly stitching these new nucleotides together, forging the sugar phosphate backbone, completing the new half of the helix. The result? 2 identical double helices, each comprising one original strand and one newly synthesized strand. This is the beauty of semi-conservative replication. Each new DNA molecule is a faithful echo of the past, carrying within it a piece of its ancestral self, ensuring that the legacy of information endures. It is life remembering itself, perfectly, precisely across the vast expanse of time. But what good is a magnificent archive, a perfect blueprint, if the instructions within it cannot be read and acted upon? The DNA, our grand scroll, generally resides safely within the nucleus of eukaryotic cells, a protected vault. It is too precious, too vast to be directly exposed to the bustling, often chaotic machinery of the cytoplasm. This is where another extraordinary molecule steps onto our stage, a close cousin to DNA, yet distinct in its dynamic roles. RNA, or ribonucleic acid. RNA acts as the crucial intermediary, the working copy, the messenger, and even the architectural scaffold for translating the genetic code into the tangible structures and functions of life. It forms the second act of what we call the central dogma of molecular biology, the elegant unidirectional flow of genetic information from DNA to RNA to protein. This dogma is not a rigid rule, but rather a profound truth about how life expresses its vast potential. The first step in this expression is transcription, the process of copying a specific gene's instructions from the DNA blueprint into a temporary RNA molecule. Imagine you have a colossal ancient cookbook, the DNA, in a grand secure library, the nucleus. You don't take the entire cookbook out to the kitchen every time you want to bake a cake. Instead, you carefully transcribe a single recipe onto a smaller, more portable note card. This note card is our messenger RNA, or RNA. Here's how it unfolds. A specific segment of the DNA double helix corresponding to a single gene temporarily unwinds. An enzyme, a polymerase, akin to a skilled scribe, moves along one of the DNA strands, reading its sequence. As it reads, it synthesizes a complementary RNA strand. There's a slight difference here. In RNA, thymine T is replaced by uracil U, so an A on the DNA template will call for a U in the RNA. Once the gene has been fully transcribed, the monomolecule detaches, a single-stranded, ephemeral whisper of the day's grand pronouncements, ready to carry its specific message out of the nucleus and into the bustling cytoplasm. And what happens to this messenger, this crucial note card of instructions? It makes its way to the cellular protein factories, the ribosomes. These complex molecular machines are themselves largely made of ribosomal RNA. Yes, and proteins. Here the process of translation begins, transforming the language of nucleotides into the language of amino acids, ultimately building the proteins that perform nearly all the work of the cell. Picture the mRNA molecule as a long ticker tape threaded through the ribosome. The instructions on this tape are read in sequential words, each word consisting of 3 consecutive nucleotides. These 3 nucleotide units are called codons. Each codon specifies a particular amino acid. This is the genetic code, a universal dictionary shared by almost all life forms on Earth, a testament to our common ancestry. But how do these amino acids arrive at the ribosome in the correct order? This is the role of transfer RNA, or tRNA. Imagine TRNA molecules as tiny molecular delivery trucks, each specifically designed to carry one particular type of amino acid. At one end, each tRNA has a unique 3-nucleotide sequence called an anticodon, which is complementary to a specific mRNA codon. As the ribosome moves along the mRNA reading each codon, the appropriate tRNA molecule, carrying its specific amino acid cargo, docks momentarily with its matching codon. One by one, the amino acids are brought into place, linked together by peptide bonds, forming a growing chain. The ribosome essentially acts as a skilled weaver, stitching together the fabric of life one amino acid thread at a time, following the precise pattern laid out by the mRNA. When the ribosome encounters a stop codon on the mRNA, the process concludes and a fully formed protein, a polypeptide chain, is released, ready to fold into its precise 3-dimensional shape and embark upon its designated function within the cell. From the silent unzipping double helix of DNA to the bustling ribosome, where a messenger RNA's code is meticulously translated into a functional protein, we witness a symphony of molecular precision. This intricate flow of information is the very bedrock of biological function, dictating everything from the color of your eyes to the efficiency of your metabolism, from the strength of your bones to the thoughts that flicker through your mind. Yet even in this system of astounding fidelity, perfect replication and transcription are not always guaranteed. Occasionally a molecular scribe makes a tiny error. A single nucleotide is swapped, deleted, or inserted. These changes, known as mutations, are often corrected by cellular repair mechanisms, but sometimes they persist. While some mutations can be detrimental, leading to disease, others are neutral, and a precious few can even be beneficial, providing the raw material for evolution, allowing life to adapt, diversify, and explore new forms and functions across the vast canvas of time. So you see, the answer to how life remembers itself is not a simple one, but a profound tapestry woven from the very fabric of existence. It is held within the elegant spiral of DNA, faithfully replicated and passed down through generations. It is transcribed into the temporary wisdom of RNA, and then translated into the tireless work of proteins. This is the quiet miracle unfolding within every cell, every moment, a continuous story of information, structure, energy, and purpose. It is the music that gives form to the dance, the script that allows the play of life to continue, an endless, awe-inspiring drama unfolding on the grandest stage of all. We have journeyed together, you and I, through the intricate architecture of life, from the silent dance of atoms yearning for stability to the grand symphony of genetic information, faithfully copied and meticulously translated. We have seen the blueprint laid bare, the very script from which existence is penned. But a blueprint, however detailed, is merely potential. A script, however profound, requires a director, a cast and a stage manager to bring it to life, to ensure that each line is delivered at the right moment by the right character with the appropriate emphasis. Consider for a moment the sheer complexity of a single cell, let alone the trillions that compose a human body. Not every gene is needed at every instant. Not every protein needs to be produced in vast quantities all the time. To do so would be a chaotic waste of energy, a cacophony instead of a symphony. How then does the cell, this miniature universe, discern when to activate a specific gene, when to silence another, or when to respond to a subtle whisper from its environment? This, dear friends, is the profound realm of gene regulation and cellular communication. The exquisite choreography that transforms static information into dynamic, responsive life. Imagine the DNA within each of your cells not as a book to be read cover to cover incessantly, but as a vast library. Within this library reside thousands upon thousands of specific instructions, each a gene for building proteins or functional RNA molecules. The cell in its wisdom does not blindly transcribe every single one of these genes. Instead, it possesses an intricate system of gatekeepers, signal flags and dimmer switches that control when, where, and how much of each gene's information is expressed. This is gene regulation, a masterclass in efficiency and precise control. At its heart, gene regulation allows cells to adapt. It enables a single fertilized egg with its identical set of DNA to differentiate into the myriad specialized cells that form a human, a neuron, a muscle cell, a skin cell, each expressing a unique subset of genes, each performing a distinct role in the grand organism. It permits a cell to respond to a sudden change in nutrient availability, to a hormone signaling from a distant gland, or to the subtle pressures of its immediate surroundings. The primary point of control, the most common dimmer switch, occurs at the level of a transcription. Before DNA can even be copied into messenger RNA, a host of molecular players must convene. Special proteins, known as transcription factors, act like molecular conductors, binding to specific DNA sequences, often upstream of a gene, to either enhance or inhibit the binding of RNA polymerase, the enzyme responsible for transcription. Think of them as the fingers on a complex control panel, fine-tuning the volume of each gene's expression. Some transcription factors are activators, turning up the gene's voice. Others are repressors, muting it. But the story of regulation doesn't end there. It is a multi-layered masterpiece. Even after an mRNA molecule is transcribed, its journey is fraught with potential control points. Its stability can be regulated. Some mRNA molecules are quickly degraded, preventing excessive protein production, while others are protected, allowing for sustained synthesis. The very act of translation at the ribosome can be modulated, ensuring that even if an mRNA is present, its message might only be translated into protein when conditions are just right. And finally, the proteins themselves, once synthesized, are subject to post-translational modifications. Subtle chemical tags that can activate, deactivate, or target them for degradation, adding yet another layer of dynamic control. We've already glimpsed this with the phosphorylation of enzymes, remember? It is a constant, exquisite dance of creation and dismantling, a ceaseless fine-tuning. What orchestrates this internal ballet of gene expression? Often it is the needs and signals from outside the cell. This brings us to the equally profound concept of cellular communication. No cell is an island, especially in multicellular organisms. They are constantly exchanging information, a ceaseless dialogue of molecular whispers and shouts that coordinates the vast cellular community. This communication allows for growth, development, tissue repair, and the coordinated response to environmental challenges. How do cells talk? They do so through a sophisticated system of signal transduction. Here, it begins with a ligand, a signaling molecule, often a hormone, a growth factor or a neurotransmitter, that acts as a molecular messenger. This ligand travels through the extracellular fluid, seeking out its designated recipient. And what is this recipient? A receptor. Protein, typically embedded within the cell's plasma membrane, specifically shaped to recognize and bind only its particular ligand. This is the ultimate lock and key mechanism, a testament to molecular specificity. When a ligand binds to its receptor, it triggers a conformational change in the receptor protein, much like a key turning a lock. This seemingly simple event is the crucial first step in a cascade of intracellular events, a molecular relay race that transmits the external signal deep into the cell's interior. This is the essence of signal transduction. Imagine a domino effect within the cell. The activated receptor then activates another molecule, which in turn activates another, and so on. These intracellular signaling pathways often involve a series of phosphorylation events where enzymes called kinases add phosphate groups to specific proteins, altering their activity. Conversely, phosphatases remove these groups, providing a crucial off switch. This phosphorylation cascade not only transmits the signal but also amplifies via it. A single ligand binding to a single receptor can ultimately lead to a massive cellular response, like a tiny ripple creating a tidal wave. The ultimate outcome of these signaling pathways is incredibly diverse. Some signals might directly activate transcription factors, leading to changes in gene expression, turning on genes that promote cell division, for example, or genes that initiate a stress response. Other signals might rapidly alter the activity of existing enzymes, changing the cell's metabolism. Still others might induce changes in cell shape, movement, or even trigger programmed cell death, known as apoptosis, a vital process for development and tissue homeostasis. Consider the elegance of this system. A single cell bombarded by a multitude of signals must interpret each one, weigh its importance, and integrate the information to produce a coherent, appropriate response. It is a constant process of sensing, interpreting, and reacting, ensuring that the cell remains in harmony with its neighbors and its environment. From the development of a complex organism from a single cell, to the daily maintenance and repair of tissues, to the swift response of your immune system to an invader, this intricate dance of regulation and communication is at play. We stand in awe of this profound coordination. The silent architects, the atoms and molecules, have built the information, and now we see the intelligent design in how that information is used, controlled, and shared. It is a testament to the universe's inherent tendency towards order, towards building ever more complex and responsive systems from simple, elegant rules. The cell, then, is not merely a collection of parts, but a dynamic, communicative entity, a living testament to the power of molecular orchestration. And as we comprehend these fundamental principles, we draw closer to understanding the very essence of what it means for life to thrive. We have journeyed through the intricate landscapes of the cell, witnessing how life's silent architects, atoms and molecules, engage in their elaborate dances, forming the very fabric of existence. We've seen how these components assemble into magnificent macromolecules, how enzymes orchestrate their ceaseless transformations, how energy flows like a vital current, and how information is inscribed, replicated, and expressed. Most recently, we stood in awe before the cell's remarkable capacity for communication, observing how whispers from the environment are translated into a symphony of internal responses, ensuring harmony and adaptation. But what, we might ask, is the ultimate purpose of this exquisite orchestration? What grand design does this intricate communication uphold? If each cell is a world unto itself, listening and responding with such precision, then what is the overarching narrative that these individual stories contribute to? The answers, dear friends, lie not in isolated events, but in the magnificent interwoven tapestry of metabolic pathways, the dynamic interconnected networks of chemical reactions that sustain life itself, Imagine, if you will, not a collection of separate streams, but a vast shimmering delta where countless rivers converge and diverge, each carrying vital nutrients, energy, and information. This is the metabolism of a cell, a ceaseless flow, a vibrant living network of transformations where the product of one reaction becomes the reactant for the next, forming an intricate web of dependencies and interactions. From the breaking down of complex molecules for energy, catabolism, to the building up of new structures, anabolism, every pathway is a thread, meticulously woven into the larger fabric, ensuring that the cell remains a self-sustaining, ever-renewing entity. Consider the journey of a simple sugar molecule, a single glucose entering the cell. It does not embark on a solitary path. Instead, it enters a bustling highway, a metabolic pathway known as glycolysis, where it is systematically broken down, releasing its stored energy. But the products of glycolysis do not simply vanish, they feed into other pathways, the citric acid cycle for instance, or perhaps into pathways that synthesize fatty acids or amino acids, should the cell require building blocks rather than immediate energy. This is the beauty of interconnectedness. A single molecule can be shunted down different branches depending on the cell's immediate needs, its environmental cues, and the signals it receives. This intricate branching and converging is not chaotic, it is exquisitely controlled. Just as a maestro guides an orchestra, ensuring that each instrument plays its part at precisely the right moment, so too are metabolic pathways regulated with breathtaking precision. Here, the lessons from our previous explorations converge. Enzymes, those alchemists of life, are the primary points of control. Their activity can be enhanced or inhibited by allosteric regulators, by covalent modifications like phosphorylation, or by the very products of the pathways they govern. This creates a system of sophisticated feedback loops, ensuring that the cell does not overproduce what it has in abundance, nor squander resources it desperately needs. Envision a negative feedback loop. As the concentration of a pathway's end product rises, it acts as a signal, binding to an enzyme earlier in the pathway and slowing down its activity. This is like a thermostat sensing that the room has reached the desired temperature and turning off the heater. Conversely, a positive feedback loop can amplify a signal, accelerating a pathway when a particular product is urgently required, much like an initial spark igniting a roaring fire. These loops are not mere mechanisms, they are the cell's innate wisdom, its capacity for self-correction and self-optimization, ensuring a dynamic equilibrium, a state we call homeostasis. Homeostasis is not a static state of inert balance like a perfectly still pond. Rather, it is a vibrant, ever-adjusting dance, like a skilled tightrope walker constantly shifting their weight to remain upright amidst subtle breezes. The cell is a master of this dynamic equilibrium, perpetually sensing its internal and external environment, recalibrating its metabolic flow to maintain optimal conditions for life. It is through these interconnected, regulated pathways that the cell maintains its identity, its integrity, and its capacity to thrive. What if we were to look closer at the sheer scale of this interconnectedness? It's not just a few pathways, it's a vast, dizzying network involving thousands of distinct reactions, all happening concurrently within the microscopic confines of a cell. Each reaction is a tiny cog, turning in perfect synchronicity with its neighbours, responding to subtle shifts in temperature, nutrient availability, pH, and an endless array of chemical signals. To truly appreciate this, we must shift our gaze from the isolated components to the magnificent orchestra they form. This is the realm of systems biology, where we seek to understand not just the individual players, but the emergent properties that arise from their complex interactions. Systems biology invites us to see the cell as more than a mere collection of molecules. It is a complex, adaptive system, capable of learning, responding, and evolving. It asks us to consider how genetic information, protein structures, metabolic fluxes, and signaling cascades all intertwine to create a living entity. It is a humble acknowledgement that the whole is indeed greater than the sum of its parts, and that the profound beauty of life often lies in these intricate, emergent properties that defy simple reduction. Consider the sheer resilience, the profound adaptability of life made possible by these dynamic networks. A sudden change in nutrient availability, a shift in temperature, the presence of a new signal, the cell does not falter. Instead, its metabolic pathways elegantly rewire themselves, shifting resources, up-regulating some enzymes and down-regulating others to navigate the new conditions. It is this extraordinary flexibility, this inherent capacity for self-organization and self-preservation that allows life to flourish in the most challenging environments, from the scorching depths of volcanic vents to the frigid poles. This capacity for adaptation, woven into the very fabric of cellular metabolism, speaks to a deeper truth about existence. Life, at its core, is a constant process of becoming, a perpetual dialogue between an organism and its environment. And this dialogue is articulated, interpreted, and responded to through the language of biochemistry, through the energetic handshakes and embraces of molecules, through the intricate feedback loops that maintain balance, and through the vast interconnected networks that allow life to persist, evolve, and ultimately to thrive. As we stand at this precipice of understanding, looking out upon the vast, interwoven landscape of life's fundamental processes, we can only marvel. We have followed the thread from the yearning of a single atom for stability to the complex, self-regulating systems that define a living cell. We have seen how simplicity gives rise to complexity, how individual interactions blossom into emergent properties, and how the silent, ceaseless dance of molecules underpins all that we know as life. But what does it mean for these systems to exist not in isolation, but within the grander architecture of a multicellular organism? How do these perfectly balanced, self-regulating cellular worlds then coordinate with billions of their kin to form a body, a mind, a consciousness? The individual cell, for all its wonder, is but a single note in the universe's grandest symphony. The next step in our journey, then, must be to transcend the cellular boundaries, to explore how these exquisite biochemical principles scale up to shape the very form and function of complex life, and how they ultimately define who we are. For the story of biochemistry is, after all, the story of us. We have journeyed through the intricate molecular landscapes, from the yearning embrace of atoms to the grand symphonies of metabolic pathways, all orchestrated to maintain the delicate balance of a single cell. But what if we were to lift our gaze, moving beyond the solitary dance of individual cellular life? What if we were to ask how these myriad, self-contained universes of biochemical activity transcend their singular existence, coming together to form something far grander, more complex, and ultimately more us? This is the profound question that Chapter 8 left whispered in the air. How do these intricate cellular systems scale up, not just to form tissues and organs, but to sculpt the very essence of multicellular organisms, to house the mysteries of consciousness, and to define that singular, irreplaceable spark we call identity? Today we embark upon the next breathtaking vista of our biochemical odyssey, exploring the astonishing leap from the individual to the collective, from a solitary cell to the magnificent integrated whole that is a living being. We shall witness the silent architects of cooperation, the subtle engineers of specialization, and the elegant process that weave billions upon billions of cells into a cohesive, breathing, thinking entity. The symphony of multicellularity, a collective dream. Imagine, if you will, a bustling metropolis, not of steel and glass, but of living, breathing cells. Each cell a tiny, self-sufficient citizen, yet bound to its neighbours by invisible threads of purpose and communication. This, dear friends, is the essence of multicellularity, a grand collaborative endeavour where the survival and flourishing of the whole depend on the harmonious interaction of its parts. It is a testament to nature's profound capacity for organisation, a quiet wonder that unfolds within us every moment. How do these individual cells, once content in their solitary existence, learn to commune, to adhere, to form the very fabric of existence? The answer lies in a sophisticated language of molecular handshakes and embraces, a vocabulary woven into the cell surface itself. We speak here of cell adhesion molecules, CEMs, the molecular glue that binds cells together, allowing them to form stable tissues. Consider the cadherins, for instance, proteins that reach out from one cell to grasp a kindred cadherin on a neighbouring cell, a calcium-dependent embrace that creates strong selective attachments. They are like the hands clasped in a dance, creating a continuous, robust connection. Then there are the integrins, molecules that not only link cells to each other, but also bridge the gap between a cell and its surrounding environment, the extracellular matrix, ECM. The extracellular matrix itself is a marvel, a dynamic, intricate network of secreted macromolecules, primarily proteins like collagen and elastin, interwoven with complex carbohydrates. It is the very scaffolding of life, providing structural support, regulating cell behavior, and serving as a crucial highway for intercellular communication. Think of it as the rich, fertile soil in which our cellular city is built, or the intricate, invisible web that supports a spider's masterpiece. Cells don't just sit on the ECM, they interact with it, pulling, pushing, sensing its stiffness and composition, receiving vital cues that dictate their growth, movement, and even their fate. Through these intricate connections of the cadherin-cadherin embraces, the integrin ECM anchors, and the vast communicative expanse of the ECM, billions of cells cease to be mere individuals and begin to coalesce into a truly collective being. They become a tissue, an organ, a unified symphony of life. Yet simply sticking together is not enough. For a multicellular organism to thrive, its cells must specialize, taking on distinct roles that contribute to the greater good. This is the profound magic of cell differentiation, a process by which cells, despite possessing identical genetic blueprints, embark on wildly divergent paths, transforming into everything from a light-sensing neuron to a sturdy bone cell, a contractile muscle fibre, or a delicate skin cell. How can the same genetic script yield such a spectacular array of cellular actors? The answer, as we have touched upon before, lies in the exquisite control of what we call gene expression. While every cell in your body contains virtually the same set of genes, it is which genes are actively transcribed and translated into proteins that defines a cell's identity and function. During development, master regulatory genes, often encoding powerful transcription factors, act as the conductors of this cellular orchestra. They don't just switch one gene on or off, they activate entire programs of gene expression, setting a cascade of events in motion that commit a cell to a particular developmental lineage. Imagine a single flick of a switch that transforms a generic blueprint into the detailed plans for a specialized engine, then another switch for a wing, and so on. Furthermore, the memory of these developmental decisions is not merely encoded in the DNA sequence itself, but also in the subtle yet profound modifications to the DNA and its associated proteins, collectively known as epigenetics. Processes like DNA methylation and histone modification act as molecular bookmarks, silencing certain genes or making others more accessible for transcription. These epigenetic marks are heritable through cell divisions, ensuring that a daughter skin cell knows it must remain a skin cell, even as it divides and replenishes the tissue. They are the subtle annotations in the margins of the genetic text, guiding the cell's interpretation of its own destiny. Cell differentiation, then, is not a random process, but a precisely choreographed dance, guided by internal genetic programs and external environmental cues, leading to the breathtaking diversity of cellular forms and functions that constitute a living organism. With specialized cells now firmly established, the next marvel unfolds, their precise self-organization into functional tissues and, subsequently, complex organs. This is where the abstract beauty of biochemical interactions translates into the tangible structures that allow us to see, to breathe, to think, to feel. How do billions of neurons know to form a brain, or countless muscle cells arrange themselves into a beating heart? This is the realm of developmental biology, a field deeply rooted in biochemical principles. Consider the role of morphogens, signaling molecules that are secreted by certain cells and diffuse through tissues, creating concentration gradients. These gradients act as molecular signposts, dictating the developmental fate of cells based on their proximity to the source. A cell exposed to a high concentration of a morphogen might differentiate into one type, while a cell further away, sensing a lower concentration, might become another. It's like a chemical compass, guiding cells to their designated positions within the developing organism. Beyond chemical signals, cells also engage in complex behaviors like cell migration, moving purposefully to specific locations, and cell sorting, where different cell types spontaneously arrange themselves into distinct layers or clusters, much like oil and water separating, but with far greater precision and purpose. These processes are driven by the dynamic interplay of cell adhesion molecules, the cytoskeleton, and signaling pathways that guide their movements and interactions. The emerging form and function of an organ, be it the intricate filtration system of a kidney or the elaborate neural networks of the brain, are not arbitrary. They are the direct, elegant consequence of these biochemical instructions, executed with astounding fidelity. We witness here the universe's inherent tendency towards self-organization, building complexity from simplicity, creating breathtaking structures from the ground up, cell by cell, interaction by interaction. In this grand drama of creation and organization, there is a paradox. Life's intricate construction often requires a controlled dismantling. This is the crucial, often overlooked role of apoptosis, or programmed cell death. Far from being a destructive force, apoptosis is an exquisitely regulated process, a necessary act of cellular self-sacrifice that is fundamental to development, tissue maintenance, and disease prevention. Imagine a sculptor refining their masterpiece, carefully chipping away excess material to reveal the elegant form beneath. Apoptosis acts as nature's sculptor. During embryonic development, for instance, The webbing between our fingers and toes is initially present, but it is precisely removed by apoptosis, shaping our distinct digits. Without this programmed cellular demise, our hands would resemble flippers. Similarly, as tadpoles mature into frogs, their tails are reabsorbed through apoptosis, providing nutrients for the developing limbs. At the biochemical level, apoptosis is executed by a family of proteases called caspases. These executioner enzymes are normally held in an inactive state, but upon receiving the appropriate signals either from within the cell, e.g. irreparable DNA damage, or from external cues, e.g. immune signals to remove infected cells, they become activated, triggering a cascade of events that systematically dismantle the cell from the inside out. The cell shrinks its DNA fragments, and it breaks into small membrane-bound packages that are then neatly engulfed by the neighboring cells or immune cells, leaving no inflammatory trace. This clean, efficient removal prevents the release of harmful cellular contents and maintains the integrity of the surrounding tissue. Apoptosis is a constant silent guardian within us, eliminating old damaged or potentially cancerous cells, ensuring the health and integrity of our tissues. It is a profound testament to the interconnectedness of life and death, demonstrating that even cessation can be a creative act, essential for the ongoing vitality of the whole. And so, we arrive at the profound culmination of our journey. We have seen atoms yearn for stability, molecules dance into complex shapes, energy flow, genetic information replicate and express, and cells communicate to specialize and organize into magnificent multicellular architectures. But what does all of this mean for you, for that singular irreducible sense of self, that unique identity that distinguishes you from every other being in the universe? From a biochemical perspective, identity is not a static blueprint, but a dynamic, ever-changing equilibrium, a continuous symphony played by trillions of cells, each following its intricate biochemical score. Your unique genetic code, the specific epigenetic modifications that have accumulated over your lifetime, the precise folding of your proteins, the specific metabolic pathways that fuel your thoughts and movements, the delicate balance of neurotransmitters that shape your emotions, all of these, in their infinite complexity and dynamic interplay, constitute the biochemical you. Consider your brain that most astonishing of organs, a universe of billions of neurons, each a biochemical marvel. It is here, in the ceaseless electrical and chemical chatter between these specialized cells, fueled by ATP, shaped by ion channels, modulated by neurotransmitters, that consciousness itself is believed to emerge. The sensation of a memory, the spark of an idea, the warmth of an emotion, these are not ethereal concepts divorced from biology. They are the emergent properties of exquisitely orchestrated biochemical events. Your identity is a narrative woven from the continuous renewal and interaction of your molecular components. Every breath you take, every meal you consume, every thought you entertain subtly reshapes the biochemical landscape of your being. You are a living testament to the elegance of self-organization, a walking, talking, dramatic marvel of biochemistry. The atoms that once were stardust, the molecules that form the primordial soup, the cells that learn to cooperate and specialize. They are all here, within you, an unbroken chain of existence, a testament to the universe's profound capacity for creation. The mystery of consciousness, the subjective experience of being you, remains one of science's grandest frontiers. Yet, we now understand that its roots are deeply embedded in the very biochemical principles we have explored. It is in the exquisite dance of ions across a neuronal membrane, the precise binding of a neurotransmitter, the intricate signaling cascades that propagate through a neural network, that the whispers of thought and feeling begin. You are, in essence, a living, breathing, profoundly complex biochemical event, a continuous, dynamic expression of the universe's most fundamental laws. This understanding does not diminish the wonder of who we are. It amplifies it, grounding our existence in the quiet, profound beauty of the molecular world. We have journeyed together through the intricate tapestries woven by life, from the fundamental yearning of atoms for stability to the grand orchestrated dance of trillions of cells forming a singular, complex identity. We have seen how the whispers of chemical bonds give rise to the robust structures of proteins, how the energetic currency of ATP fuels every cellular endeavour, and how the silent language of DNA dictates the very blueprint of existence. We have marvelled at the exquisite precision of enzymatic catalysis, the elegant flow of genetic information, and the sophisticated conversations that unfold between cells, shaping their destinies and building the very tissues and organs that allow us to perceive and interact with our world. Now, we stand at the precipice of the most profound mystery of all, a frontier where biochemistry, for all its explanatory power, meets the ineffable. We have explored how individual identity emerges from the biochemical symphony of a multicellular organism, how the coordinated actions of specialized cells give rise to a cohesive living being. But what of consciousness itself? What of the subjective experience, the inner world that makes each of us a unique universe of thought, feeling, and perception? How does the ceaseless, intricate ballet of molecules, ions, and electrical impulses within the brain give rise to the vibrant, shimmering reality we call being? Consider for a moment the sheer audacity of this question. We have meticulously dissected the mechanisms of life, revealing the elegant logic of its construction. We understand the neurotransmitters that bridge synaptic gaps, the ion channels that propagate electrical signals, the metabolic pathways that fuel neural activity. We can observe the brain's intricate architecture, map its regions, and even track the flow of information as it processes sensory input or generates a thought. Yet the leap from these observable, measurable biochemical events to the feeling of seeing a sunset, the experience of love, or the awareness of one's own existence remains the ultimate enigma. We are not merely a collection of reacting chemicals, are we? Or are we? This is where the scientific narrative, for all its brilliance, bows to a deeper, more philosophical inquiry. Biochemistry provides the canvas, the paints, the brushes, and even the laws of physics that govern their interaction. But who is the artist, and what is the masterpiece being created? We can trace the path of a photon striking the retina, triggering a cascade of biochemical events in photoreceptor cells, leading to electrical signals that travel along the optic nerve, through various nuclei, and ultimately to the visual cortex. We can even pinpoint the specific neural networks that activate when you recognize a familiar face. But where in this intricate biochemical cascade does the experience of seeing red or the feeling of recognition truly reside? This is the hard problem of consciousness, a challenge that transcends the boundaries of any single scientific discipline. Yet it is within the realm of biochemistry that its most fundamental underpinnings are laid bare. The brain, that most wondrous of organs, is perhaps the most biochemically active and complex structure in the known universe. It is a dense, pulsating nexus of trillions of synapses, each a tiny biochemical theatre where neurotransmitters are released, bind to receptors, and trigger new cascades of intracellular events. Think of the neurotransmitters themselves: serotonin, dopamine, acetylcholine, GABA, glutamate. These are not abstract concepts, they are tangible molecules, crafted with exquisite precision, each with a specific shape, a specific affinity for its receptor, and a specific role in modulating neural activity. They are the chemical words in the brain's ceaseless conversation, dictating mood, memory, learning and motor control. A subtle shift in their concentration, a slight alteration in their receptor binding, can profoundly reshape our perception, our emotions, our very sense of self. When we learn something new, when we form a memory, what is happening at the biochemical level? We are witnessing synaptic plasticity, the strengthening or weakening of connections between neurons. This involves the insertion or removal of receptor proteins, changes in gene expression within the neuron, and the remodeling of dendritic spines, all processes dictated by the elegant choreography of biochemistry. Our memories are not stored as static files, but as dynamic, ever-changing patterns of biochemical potential, ready to be reactivated and re-experienced. And what of the electrical impulses, the action potentials that zip across neurons at astonishing speeds? These are not purely electrical phenomena, they are fundamentally biochemical. The opening and closing of voltage-gated ion channels, complex protein machines embedded in the neuronal membrane, allow the rapid influx and efflux of ions like sodium and potassium, momentarily reversing the membrane's electrical potential. This precise timed movement of charged particles, driven by protein pumps and channels, is the very heartbeat of neural communication, the rhythm upon which all thought and experience are built. The brain's insatiable demand for energy is another testament to its biochemical intensity. It consumes a disproportionate amount of the body's ATP, constantly fueling ion pumps to maintain membrane potentials, synthesizing neurotransmitters, and building new proteins for synaptic remodeling. Every thought, every feeling, every flicker of awareness is powered by the relentless combustion of glucose transformed into ATP through the intricate pathways of cellular respiration we explored previously. Consciousness, then, is an energetically expensive endeavor, a testament to the sheer biochemical labor involved in maintaining this state of active, reflective awareness. Yet, even as we dissect these marvelous biochemical components, the question persists. How do these individual reactions, these molecular interactions, coalesce into the subjective, unified experience of you? It is not as if a single molecule of serotonin is happiness or a burst of dopamine is reward. Rather, it is the pattern of their release, the network of their interactions, the emergent property of billions of neurons firing in concert that gives rise to these complex states. Imagine a grand orchestra composed not of instruments but of molecules. Each molecule plays its part, a specific note or rhythm. The neurotransmitters are the melodies, the ion channels the percussive beats, the energy molecules the very breath of the musicians. We can analyze each instrument, each note, each musician's technique. But the symphony itself, the emotional resonance, the cohesive beauty that moves us, that is more than the sum of its parts. Consciousness, in this sense, is the ultimate biochemical symphony, a ceaseless, evolving composition played out on the stage of the brain. It is a profound humility that biochemistry teaches us in the face of this ultimate mystery. We can map the pathways, identify the players, and even manipulate the script with various pharmacological agents, altering mood, perception, and thought. Yet, the qualia, the raw subjective feel of an experience, remains stubbornly elusive. We understand the how of the brain's operation, but the what it is like to be conscious, to feel and know and be, continues to beckon us from beyond the molecular veil. Perhaps the beauty of this journey lies not in finding a definitive answer, but in the very act of asking the question. To ponder consciousness from a biochemical perspective is to reflect on the universe reflecting on itself. It is to recognize that the very atoms forged in distant stars, the very molecules that dance in the cosmic dust, have, through billions of years of intricate self-organization, coalesced into systems capable of contemplating their own existence. We are, in a very real sense, the universe made manifest, capable of observing and understanding its own biochemical language. And as we stand here, at the threshold of this grand unknown, we are reminded that the journey of discovery is far from over. Biochemistry, with its quiet wonders and profound beauty, continues to illuminate the path, revealing layer after layer of the intricate, elegant mechanisms that underpin life, identity, and perhaps one day, even the deepest secrets of consciousness itself. The questions are as beautiful as the answers, and the ongoing exploration is the greatest adventure of all.